Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled ALS Biomarkers in Drug Development. My name is Sydney, and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank ARG, who developed the content for this presentation. ARG is an oncology, immunology, rare and neurodegenerative disease-focused contract research organization that provides comprehensive clinical program development services ranging from pre-launch consulting to commercialization, achieving over 50 market authorizations. Founded in 2004 with the vision that every project should be highly individualized, ARG has experienced consistent growth across the globe, expanding its reach to include drug and device strategic consulting, clinical trial management services, and clinical data and analytic solutions. ARG uses first-in-class technology platforms along with relationship-driven flexibility to optimize clinical studies because it believes everyone deserves to be well. And now I'd like to introduce our speakers for today's event. Dr. Ignazio di Giovanna, ARG Vice President of Europe, has deep experience in the clinical research domain, having worked in the CRO field since 1990 and the pharmaceutical industry as a whole since 1986. Previously, Ignazio owned and operated the European CRO CCA Clinical Research from 1999 until its acquisition by ARG in 2018. Ignazio earned a PhD in biophysics at Guy's Hospital based on the development of non-invasive cardiovascular diagnosis techniques. Ignazio has been involved in neurodegenerative disease drug development with a focus on ALS for over 10 years. Dr. Toby Ferguson is a neuromuscular neurologist and neuroscientist who joined Biogen in 2013. His professional experience has been focused on developing treatments for neuromuscular disease with a focus on ALS. He is currently the head of the Neuromuscular Development Unit at Biogen and leads the Neuromuscular Clinical Development Group. Toby plays a key role in developing ALS clinical trials and in driving preclinical strategy with neurodegenerative and neuromuscular disease. Prior to Biogen, Toby had a clinical neuromuscular neurology practice and a lab focused on peripheral axon injury and regeneration at Shriners Research Center and Temple University in Philadelphia. Toby trained in neurology and neuromuscular disease at the University of Pennsylvania. He obtained an MD and PhD in neuroscience at the University of Florida and maintains an interest in axon regeneration and degeneration. Dr. Angela Genge has been the director of the Clinical Research Unit at the Montreal Neurological Institute since 2004. Her career is notable for her groundbreaking research, prominent leadership positions, and significant distinctions, particularly in neurodegenerative diseases. Angela's research focuses on drug development in rare neurological diseases, and she is known for clinical and clinical trial design expertise, as well as the introduction of innovative therapies. She led programs in the ALS, FTD, genetic forms of myopathies, and myasthenia gravis. Angela completed her medical degree at the Memorial University of Newfoundland. She completed her Canadian and American certifications in internal medicine and neurology prior to completing a fellowship in neuromuscular diseases. She joined the staff of the Montreal Neurological Hospital in 1994 and became director of the ALS clinic in 1998. Angela has distinguished herself in her career, receiving the Forbes Norris Award, the Diva of Distinction Award, and the Governor General Diamond Jubilee Award. Now, before passing controls over to our speakers, I'd like to introduce our webinar for today. In a few quick words, this webinar is about the use and development of innovative biomarkers in ALS clinical trials. And now, without further ado, I'd like to hand the mic over to our first speaker, Ignazio. You may begin when ready.
Well, thank you, Sydney. As we have a number of attendees from various time zones, I'll simply say good day and welcome everybody. I just wanted to take a few moments to do a very brief overview of ALS and set the scene as to why we're going to be concentrating on ALS biomarkers for this talk. The symptoms of what we now know as amyotrophic lateral sclerosis, or ALS, were first identified in 1869 by the French neurologist Jean-Martin Charcot. Amyotrophic is from the Greek, meaning no. Myo refers to the muscle, and trophic, nourishment. So effectively, no muscle nourishment. Lateral indicates the area in the spinal cord where the portion of the nerve that controls muscles are located. And as this area degenerates, it leads to scarring, hardening, i.e. sclerosis in that region. ALS is also known as Lou Gehrig's disease after it ended the career of Lou Gehrig, Major League Baseball player for the New York Yankees back in the early 1940s. But there have been many notable victims of ALS, unfortunately, amongst them Mao Zedong and, of course, Prof. Stephen Hawking. ALS is also known as motor neuron disease. In general, about 90% of ALS cases are sporadic, i.e. there is no one obvious cause of the disease, although many environmental factors have been implicated, such as environmental toxins, viruses, radiation, and electromagnetic fields. Whereas 10% of ALS is termed genetic or familial, in which more than 30 different gene mutations have been linked to ALS, namely C9 or 72 and SOD1 genes. ALS is regrettably a neurodegenerative disorder characterized by the loss of motor neurons, the nerve cells that control voluntary muscle movement. ALS is marked by symptoms that include muscle wasting, therefore, atrophy, and difficulty in breathing and going about everyday tasks such as dressing and eating. Around 2/3 of ALS have what is called limb onset form of disease. Initial symptoms of this form include muscle weakness and wasting in the arms and legs, and involuntary muscle contractions resulting in twitches. Early signs of this form of ALS include loss of balance, trouble walking, with constant tripping or stumbling, and difficulty performing tasks requiring fine dexterity. Patients with limb onset ALS tend to progress to paralysis and or death within 3 to 5 years. In the bulbar onset form of ALS, the muscles involved in speaking, swallowing, and breathing are generally the first to be affected. Initial symptoms therefore include slurry speech, difficulty in swallowing. However, muscle weakness can quickly progress to arms and legs, making it difficult at times to distinguish between limb and bulbar onset ALS. Bulbar ALS is slightly more common in women and people over the age of 70. Although the progression of the disease is variable, bulbar ALS tends to be faster progressing than limb onset, and patients can be paralyzed and progress to respiratory failure within 2 to 3 years of onset. Eventually, regrettably, neuronal death and loss of control of voluntary muscles leads to significant muscle weakness and eventually complete paralysis and death, primarily due to respiratory complications. Next slide, please. So just to look at some very basic disease statistics here, there's a slightly higher incidence in Europe than in North America, with generally between 7 and 15,000 new cases per year. Prevalence is also slightly higher in Europe, resulting in between 16 and 40,000 people living with the disease at any one time. Importantly, disease presence is anticipated to rise significantly by up to 70% in the next 25 years. And this is primarily really due to our increasing aging population. In general, ALS is more prevalent in white population over the age of 40, and more so in males and females. In males and females up to the age of 65, after which it tends to even out. Crucially, and unfortunately, there is no cure for ALS at this time. Next slide, please. So we have some real challenges in ALS, really. Diagnosis can be very difficult and lengthy, and it can take 12 months or sometimes longer to diagnose, and involves the need to eliminate other possible conditions in many situations. And given that the median survival post-diagnosis is in the region of 3 years, the diagnostic period really does represent a fairly significant portion of a patient's life expectancy. In terms of clinical trials, there have been a significant number of clinical trials performed on ALS. In fact, there's been over 80 randomized clinical trials published since the 1980s, but unfortunately, very few of these trials have returned any positive results. And to date, despite all this effort, only 2 drugs have been approved for treatment of ALS. Riluzole was the first in 1995, followed by edaravone quite significantly later on in 2017. So I think this shows that there is a very, very strong need for additional research understanding for potential treatments in ALS. And the webinar here, therefore, concentrates on ALS biomarkers. Next slide, please. So what is a biomarker? Well, a biomarker is simply a measurable indicator of some biological disease process, and it's usually used to define normal acceptable ranges or chart response and progress of some kind of intervention. And we're all very familiar with biomarkers every day, just looking at blood tests, for example. So although biomarkers in themselves aren't new, there is an increasing focus and interest by academia, by biotech and by pharmaceutical companies in developing and applying novel biomarkers to assist in target drug development, to assist with regulatory decision-making, and perhaps most significantly in selecting the most appropriate biomarker when looking at a drug's specific mode of action. So biomarkers can therefore provide a very useful tool to both define patient population as well as to follow a drug's effectiveness. And we're very much hoping that developing biomarkers will assist in clinical research and drug development in general. So with that, I'll hand over to Toby. Toby, over to you.
Thank you, Ignazio. Next slide, please. So I want to take a few moments and talk about the potential uses of biomarkers throughout the clinical development course. And then Angela and I will review some of the current biomarkers as examples of this. So it's important to note that throughout the process of drug development, what you see is sort of outlined in the blue middle bucket, sort of starting with identification of your target, sort of refinement of your target, identifying molecules then can affect that target, your preclinical studies, your tox studies, and then your formal clinical development phase 1, 2, and 3, And then also even after approval of a drug, there can still be uses of a biomarker. So really, preclinically, before you get into the clinic, in your cell models or your animal models and your toxicology studies, you really want to know, has the experimental compound that's been developed, is it really starting to engage the appropriate mechanism of action, engage the target? Does your drug even have legs? And so as you work through this, this is usually initially done maybe in cells and then maybe in disease models. And so you're really thinking about biomarkers really should start very early in the drug development process and not wait for the clinic, for example. And then as you successfully identify a compound, you get some data in cells and early models, you think that it's moving forward appropriately. Then the next steps are within your preclinical safety assessments is again asking the question in your safety models, are you appropriately engaging the target? So if your drug, for example, modulates excitotoxicity, can you have evidence that it's getting into the tissues you need, say the brain, in the case of ALS, in the brain and spinal cord, and then modifying that mechanism of action. You then sort of think through your dose selection, and so preclinically you use your models, and you then scale that to your projected human doses, and so all this is quite important preclinically. And then as you transition into phase 1, again, you're really initially going to ask yourself, through measurement of target engagement, through potential measurement of downstream pharmacodynamic measures, and then of course for safety, is your compound safe in phase 1? And then are you able to show that you can get appropriate dose ranging and target engagement in early phase 1? And then as you move onward throughout the clinical development course, you're really starting to ask slightly different questions. I think the safety question will persist throughout clinical development, but in phase 2 and phase 3 in particular, you're really asking, you're starting to move towards traditionally a population of patients who have the disease of interest, in this case ALS. So that case, can a biomarker help you pick the right populations? Neurologists generally think of ALS as one type of disease, but we know there are certain genetic subtypes. And we'll give you some examples later in the talk today of how genetics can really help you pick the right population. And that can be particularly important if you think of a trial design in which you included all comers, but you had a drug that only worked for, say, 20% of that population, your chances of success would be quite small. So really, patient stratification or patient enrichment is critical, particularly as we think about precision medicines. And then always you are starting to ask, once you've identified that right population, can you then start to get indications of a treatment response? And really that comes at a couple levels. One, do you see evidence of your target being engaged in your population of interest? And then two, are there prognostic markers? Can you really start to see an early indicator of efficacy? And this is sort of the holy grail within ALS. I don't think we're there yet, but I'll review a bit of data on a molecule called neurofilament, where this molecule may serve this purpose, but there remains work to be done to really formally show this. I think at the end of the day, what we really need are biomarkers that hopefully change before the clinical measures we use in the trials to really get that early read on efficacy. If those kind of tools can be identified, and we can understand the relationship of a biomarker changing in patients to predictions for efficacy, that really will enable us to make very crisp decisions without having to do full clinical trials. So that's why that's sort of considered the holy grail, and why there's so much interest within biotech, within pharma, and with academics to really better inform drug development, make it more efficient, and help us to accelerate molecules that look promising and stop molecules that don't look promising. But again, as I said, there's really sort of limited availability of such validated biomarkers, but there's active interest in developing such. Next slide, please. So I think here we're going to do some poll questions.
All right, thank you very much. Yes, we do have a poll question for our audience today. So our poll question is, what barriers are preventing the use of biomarkers in ALS clinical trials? And now our options are, I question the investment and value versus effort. I don't want to obtain regulatory validation. I am unclear about their usefulness. I am wary of their levels of validation, or I don't want to complicate the protocol. So I'll just give our audience another couple of seconds to get in their votes. And the question again is, what biomarkers are preventing the use What barriers are preventing the use of biomarkers in ALS clinical trials? I question the investment in value versus effort. I don't want to obtain regulatory validation. I am unclear about their usefulness. I am wary of their levels of validation, or I don't want to complicate the protocol. So I'll just give our audience another couple of seconds to get in their votes, and then I will close the poll and display our results. All right, it looks like we have a majority that have voted, so I'm going to close the poll and share our results. Okay, so for our poll question, we have 7% for I question the investment and value versus effort, 5% for I don't want to obtain regulatory validation, 29% I am unclear about their usefulness, 53% I am wary of their levels of validation, and 7% I don't want to complicate the protocol. So thank you very much for our audience for voting in this poll question. Back to you, Toby.
Thank you, Sydney. So maybe a couple quick comments on the poll. I'm pleased to see that folks don't question the usefulness, but I'm cognizant of the highlighting of the need for validation, and maybe we can hit on some of these topics as we go through the summary of some key biomarkers in the next couple of slides. So biomarkers come in all types and flavors. As Ignazio highlighted, they're not new, and what you can see here are a number of different examples. And really, they come in buckets of things you can measure in fluids. That can be in blood, it can be in CSF, it can be in a piece of tissue. And these are the most common, the easiest to implement, and really try to get after certain biological processes accessible in those matrices. There are plenty of other non-fluid-based biomarkers, and those we'll talk about a couple of those. Various imaging modalities such as PET or MRI, and then also electrophysiologic measures, particularly important in the nervous system and in ALS. We'll comment on a few of those. And there's also the emerging digital space. We won't spend much time on it. It's a separate topic. But in general, you can measure biological processes in many different ways. And the simplest way to think about it is anything in which you can measure a biological process could be a biomarker. And so next, what we'll do is I'm going to go through a few slides, and then I'll alternate with Angela. We'll have a bit of a back and forth, and then we'll open up for discussion as we move through the talk. Next slide, please. So in my initial schema, I highlighted the importance of asking yourself the importance of genetics, and then the importance of identification of a certain population, and the importance of target engagement. And so what you can see here is an example of some data that we've recently published around the SOD1 ALS trial. And so just to give a bit of background, this is a trial in which we're looking at a subset of ALS patients with mutations in SOD1, the SOD1 gene. We know that gene is causative of their ALS. The medication being developed for the potential treatment of SOD1 ALS is called tofersen, and this is an ASO that inhibits the production of SOD1 mRNA, therefore decreases the amount of, or the intent of it is to decrease the amount of SOD1 production. And we know based on the biology of the disease that the SOD1 molecule itself is toxic and drives the progression of the disease. So the overall intent of this molecule is to lower SOD1 and hopefully do something important clinically for these patients. And so in that way, this is a nice example of a population that's been selected by genetics. So we stratified the trial based on the genetics of the patients, and that's driven by the population selection. But here what you can see is some very nice data that asks a very simple but very fundamental question. Does the molecule, when you put it in people, do what we think it should be doing? And that really is, the more succinct form of that question is, does the tofersen ASO reduce SOD1 when you put it into the spinal fluid? I'll just highlight that in this case, what patients receive an injection of the ASO into the spinal fluid, and then we measure this CSF SOD1 over time and ask, does it decrease? And what you can see, particularly at the highest dose, 100 milligrams, that in the short early safety study and PK study, that you do see substantial reduction of SOD1 of about 40%. And so really, the point here is that in this context, this molecule is doing early job, and its job is to reduce SOD1 in the CSF. So this gives us confidence that the SOD1 is also being reduced in the spinal cord and the brain, and so it's an early signal based on a biomarker showing that we see reduction of SOD1. And so it's an early encouraging signal. It's one of the signals that made the team move forward and set up a phase 3 trial for this molecule. So again, the broader point here on neurofilament is that, on SOD1 reduction, is that it's a measure of target engagement, and that's a crucial early step in the overall drug development process. Next slide, please. So I've talked a lot about, in my schema up front, I talked a lot about the importance of biomarkers that could help us think about treatment response. And if we could identify those biomarkers, it would be really crucial. I also highlighted that we don't really have those markers yet. So I did want to talk a little bit of time on one marker that has some potential in this regard. It's been an area of active research throughout the ALS community and throughout other neurodegenerative diseases, and that is neurofilament. Neurofilament is an intermediate filament found in axons throughout the nervous system. And the overall general hypothesis, which you see schematized here on the left, is that when axons die, they release this neurofilament, and it can be detected in the CSF, and it can be detected in the blood. From a working hypothesis and importance to clinical trials, you could then hypothesize potentially that if neurofilament represents ongoing axon injury, perhaps if you reduce that axon injury, neurofilament will go down. In addition, if you wanted to think about finding patients who have evidence of that axon injury, you could pick patients for enrollment in a trial who only have elevated neurofilament. Next slide, please. So what we simply know about ALS is that if you measure neurofilament, either phosphorylated neurofilament heavy, which is on the left, or neurofilament light, which is on the right, what you can see is that compared to disease controls, neurofilament is quite elevated in ALS. And this is not by no means unique to ALS, but it's certainly clinically commonly observed in clinical patients that they do have elevated neurofilament. And it's important to note you can measure it in blood, which makes this a relatively simple and straightforward thing to do in a clinical trial. Next slide, please. So here we have an example of the same trial I just referred to, the SOD1 ALS trial. And in that trial, what's been done is the neurofilament has been measured over time, and it's been measured in plasma and CSF, both neurofilament light and neurofilament heavy. The neurofilament heavy are the two panels on the left, and the two panels on the right are neurofilament light. Overall, in addition, what you see on the top and the bottom panels is it's the overall population on the top panel, and it's the faster patients on the bottom panel. And what you can see, particularly if you look at the faster progressing patients in this short period of time, is that there seems to be the emergence of reduction of neurofilament, both neurofilament heavy and neurofilament light in plasma and in CSF, that has emerged over time. In addition, what was observed in this trial is potential trends on clinical signals. And so this is one of the few examples in ALS of a potential treatment response based on neurofilament. And so here what I'm highlighting is if we can do more research and better understand this response, it may be a clear tool to help us select important molecules to move forward in drug development for ALS. That being said, there are quite a few unknowns, and I'll highlight a couple of those unknowns. One is reduction of neurofilament indicative or formally required for efficacy in all forms of ALS or all disease mechanisms we tend to treat. We don't know that. Is how much should you reduce neurofilament for there to be a clinical effect? We don't know that. We would love to understand that decrement and understand the clinical effect that decrement implies. That remains a key unknown. Are there confounders that would make a neurofilament not change? For example, safety signals. And so here, this is some promising early data. It suggests neurofilament could be a marker of treatment response. It may help us pick molecules to move forward and aggressively move forward in drug development, but the research is by no means complete. Next slide, please. So I think I'm going to turn it over to Angela now to talk a bit about oxidative stress.
Thanks, Toby. And I'm going to start with an area that's far less validated than what Toby covered, but equally important for our future. I think Toby's comments and presentation really highlight the positive relationship between drug development in industry and academia, because both what Toby presented and the history behind that took at least 15 groups to develop the data set that's led to the use of neurofilaments in ALS clinical trials. Now let's switch hats to another area that has generated a lot of activity, and in fact has been a part of drug development for ALS for a long time. That is oxidative stress. We have not yet come to a data set that really guides us as to which marker of oxidative stress will be most informative in clinical trials in ALS. And I must say that the emphasis of this aspect of biomarker development is really around that phase 2 proof of concept range. It's wonderful if you have it preclinically and in your phase 1, but it makes its biggest difference in your phase 2 programs. And in oxidative stress, in order to move this area forward, there is currently an ongoing study that is being led by the NEALS, or the Northeast ALS Consortia, called REFINE, that is actually directly looking at a number of oxidative stress markers. The ones listed on this slide are all within the boundaries of that particular program, which is a longitudinal biomarker program. And of these, the one that has moved farthest along in clinical trials has 3-nitrotyrosine, or NT3, 3NT, and we are really awaiting this result from this REFINE program led by Dr. James Barry to really guide us into how to integrate these markers in the current round of assets that are being directed towards oxidative stress in ALS. It's a fundamental concept, but we still do not have a biomarker that stands out as fully validated for those of you who are in that 53% of worried about the weight of validation of a biomarker. Next slide, please. I am more hopeful in the area of neuroinflammation. Neuroinflammation was a target of ALS drug development way back before Toby and I were doing this work, which is a long time ago now. And in fact, it went into a very quiet stage. However, in the last 3 years, there's been a huge resurgence of interest based on a variety of programs that neuroinflammation is key in the loss of motor neuron function in ALS. What has come out of a couple of really important networks is that the CHIT levels may be important, and maybe the macrophage-derived biomarkers that are listed here may in fact be very useful in the long term as a marker of neuroinflammation. There are also several programs looking directly at complement activity, which has been studied in a number of other rare diseases, which gives us a leg up in some ways in the validation of a biomarker. There are at least 3 programs currently in development that are in clinic that are looking at complement activity. We will be borrowing from the autoimmune space to really determine which of those assessments is most accurate. Finally, I want to mention under neuroinflammation, an area that sort of crosses the boundaries between neuroinflammation, neurodegeneration, and that is muscle biopsy. Muscle biopsy is, as I like to call it, a renewable resource. In that a muscle biopsy can be repeated. Muscle biopsies are now being used in the rare muscle diseases, such as facioscapulohumeral dystrophy, to actually follow target engagement and follow effect of new assets, and is showing some promise in that field. Most recently, there has been direct work by a number of researchers, including people in the US, Canada, and Europe, looking at muscle biopsy techniques, particularly biopsies of the neuromuscular junction, as a potential biomarker in early phases of clinical trials. We know muscle biopsies can be used in pivotal phase 3 in rare disease, but its usefulness in phase 2 or proof of concept may actually be far greater than we had previously understood. So I think that watching the biomarkers of neuroinflammation that mentioned here, particularly the macrophage-derived biomarkers and the complement activity, we should be seeing some confirmation of these discoveries within the next 2 years. Next slide, please. Over to you, Toby.
Thank you, Angela. So this is a slide meant to be an exemplar of another genetic approach to disease. BIP78 is an ASO that we're developing for the treatment of the largest genetic population of ALS, C9 orth ALS. Briefly here, it's really an example in the biomarker realm. On the left, you can see schematized the cause of the disease, and that's an expansion repeat within the intron between 1A and 1B intron of this gene. And that expansion repeat causes either repeat-mediated or toxicity from the RNA structures, or there's productions of toxic peptides from that RNA. And it's those two mechanisms are thought to drive the disease, the progression of ALS. And so from a biomarker point of view, what's been done in this program is that we've developed assays that can measure those toxic species, and then you can ask the simple question, does the molecule itself reduce those toxic species? And on the right, you can see examples, at least in mice, and we'll measure this in people as the trial progresses, measurements of poly-GA and GP. These are the two examples of these toxic peptides. And what you can see in mice is that these are indeed reduced. So really, in terms of biomarkers, this trial gets us two things. One, we only study this ASO in the C9 orth population, so we've used genetic testing to stratify the patient population. And then because of the knowledge of the biology of the disease, we've identified an assay that measures the toxic species in this disease and asks, can they be reduced? The relationship of GA and GP to toxicity and to clinical effect, we don't have that fine-grained knowledge, but of course, if we weren't able to reduce these GA and GP, for example, we would have a lot of concern about the drug being able to be effective. With that, I'll turn it back to Angela for some comments on miRNA.
Next slide, please. So miRNAs or microRNAs, these are small non-coding RNAs that regulate the gene expression through splicing and silencing of mRNAs. What's important is that they are becoming increasingly recognized as markers of neurodegeneration and therefore, as which is important in this talk, potential biomarkers in drug development. The original information came out of pathological or post-mortem spinal cord specimens, but we are in a very different era now. There is a strong group led by Hornstein in Israel and another strong group led by Pam Shaw in England, as well as several American groups that have been looking directly at the importance of dicer activity that is decreased in ALS models. And currently, a number of different groups have identified either upregulation or downregulation of different microRNAs. The challenge we have, and this applies not only to ALS, but also FTD, because one of the pivotal studies came out of Toronto, Dr. Janice Robertson's group, where she worked on the Genge collection of CSF in FTD and frontotemporal dementia, and found that, in fact, we have a downregulation of certain microRNAs in that cohort. Dr. Shaw's group with Dr. Waller have actually identified increases in certain microRNAs. Originally, Dr. Shaw's group actually demonstrated the changes in the CSF, but very importantly, we now have the ability to demonstrate these changes in sera. And as we all know, as wonderful as CSF is and quite precious, it is advantageous to develop some of these assays in the sera, particularly with the breakthrough in Simoa technology. So what we expect from the various groups working on this, and we hope that these options are integrated in some of these development programs, that within, again, a couple of years, we will land on which microRNAs are the best in terms of being reliable, being able to distinguish patients with ALS from normal controls, ALS mimics, and other categories of neurodegeneration. With the information we have to date, the strongest evidence lies with the serum levels of microRNA 206 and 143 out of the Sheffield group. But until we have further validation, that group was able to validate these in their own cohort and an additional cohort. We do need further validation of these biomarkers. It is also important to remind you all that these are not free-floating, that typically the microRNAs are concentrated in exosomes. So a lot of this work is using a combination of technology to identify the most appropriate microRNA to follow in longitudinally in our ALS programs. Over to you, Toby.
Thank you, Angela. So next slide, please, Sydney. Here, I just want to make a couple comments and remind the group that there are a cluster of outcome measures that can be used as biomarkers, sort of roughly grouped as electrophysiology. The most common ones are simple EMGs, which are done routinely in clinical practice, and these can measure compound motor action potential. On the left, you see a motor neuron with its axons connecting to a muscle. That's the fundamental structural unit within the spinal cord to muscle that allows you to function. It's degeneration of that motor neuron that is key to the process of ALS. With electrophysiological markers, particularly CMAP, what you measure is the motor action potential that's generated when you stimulate the nerve and record a response in the muscle. So that sort of gets at the heart of the biology of the disease, and that's why this is attractive. On the right, you can see this is a technique called Munix, which uses a combination of CMAP and some other additional techniques to try to estimate motor unit number, so the number of motor neurons and their connection to muscle. And the disease process over time that declines. And what you can see is the top line is the ALS clinical rating scale, and the bottom line is Munix, and that declines a bit more quickly. And so the electrophysiologic measures, Munix and CMAP, really potentially offers a direct insight to the biology of the disease. And so they could be quite important to measure. I would be remiss in highlighting that there are a number of technical challenges to do this well, to do it consistently, particularly as you think about doing this within the context of a broad clinical trial where many different sites would participate, and you really have to work to get the technical aspects of this to move forward, but it really could offer important insights to the disease. Back to you, Angela.
Next slide, Sydney, please. Thanks, Toby. I think we'll see a lot more of neurophysiology, a resurgence there. And here is not the least, but the most exciting area, I believe, and that is imaging in ALS. The imaging challenge in ALS has not been through lack of effort, but it has to do with a couple of key pieces of information. One, ALS patients lose the ability to lie flat over time. So any imaging acquisition that requires more than 15 minutes becomes a problem as the disease progresses. As we all know, respiratory insufficiency is a big part of ALS. And so although we were able to break through with MS in the 90s because of MRI, we have been challenged in ALS to find imaging techniques that can be completed in a very short period of time so that they can be performed longitudinally and be used in a development program. Currently, PET imaging, particularly carbon-11 PBR28 PET, has been used to really identify areas, particularly in the prefrontal cortex, of change in patients with ALS and distinguishes ALS patients from normal controls. The PET imaging has not been used extensively. It has been used in some early programs. But what is most interesting is that PET has been correlated with two other potentially even more effective techniques, diffusion tensor imaging and magnetic resonance spectroscopy using single voxel, which between the three of them may actually allow us to move certain molecules forward. One of the things that is happening in the field of dementia, obviously, is using ligands that target amyloid and tau, and it's become particularly effective in some recent programs in identifying a target engagement and actually improvement at the level of the imaging. There are research groups internationally working on more ALS-specific ligands that will target areas that can be changed with our effective molecules. But the opportunity that some of the new PET, DTI, and MRS technology is giving us, particularly the single voxel, may actually be extremely important in our early development programs. Next slide, please. So with that, we are at the discussion period. I'm going to let Sydney or Ignazio host us for a little bit.
Thank you. While we're waiting for some questions, perhaps I can start some discussion off by just asking your opinions on where are the gaps, do you think, in terms of use and utility of biomarkers?
Maybe I'll start with, I think, the one that I've mentioned, but I'll emphasize and highlight it because my perspective is sort of the clinical development process. It's really developing a biomarker that we think captures treatment response, and we understand the predictivity of that response based on, for example, say neurofilament, to a clinical effect, and really making that biomarker to clinical effect relationship is the biggest gap that I would love to see solved. I think the other one I'll highlight is in ALS, for example. We've talked a lot about neurofilament. We haven't talked about, for example, TP43, which is the key pathology in the most patients with the disease, but we don't have accurate ways as of yet to measure that pathology, either in the CSF or blood, or through PET imaging, for example. And there's active efforts to develop those tools, but they're not ready for prime time yet. And so I think broadly, things I've highlighted are really ways to access the fundamental pathology of the disease, not always there yet. Neurofilament's exciting, but it's downstream of that. And then also tools to predict clinical response.
So I would add in, Toby, I completely agree with you. I would add in one other gap, and it's a what I call a functional gap. I highlighted what has been coming out of some of the academic labs. The gap for me is between what is being discovered in the academic lab and what can be applied in our development processes. We have a number of really, really interesting assets being moved through development, targeting all of the pathways that we've mentioned. There are academic groups that have developed techniques that, at least on their first pass, has a very high utility in some of these programs. The challenge is that second level of validation, either in a new population of patients or in a larger population of patients, needs some help in terms of being a reality. There is a number of calls that are working on this now. I believe following the imaging space is really, really important. I agree with Toby, we don't have a direct marker of that TDP43 pathology. So I think that is effort is super important, given how common that appears to be as an effect in ALS, and moving some of these other serum-based biomarkers forward, simply because the opportunity to sample serum is much more frequent than the opportunity to sample CSF.
Thank you. Thank you very much for that. And what do you think are the key questions and areas for opportunities moving forward? Angela, do you want to say that first?
So I think we're circling back to the same question. My question is Toby's question in a way. What is the best biomarker as an indicator for clinical effectiveness or efficacy. So there are two different kinds of biomarkers in my mind. Those that portend a clinical benefit, and those that are really very specific to ensuring that you have target engagement. They can be one and the same, but they can be different. So I would really like to see further work to see if neurofilament is that a biomarker indicating efficacy, or whether there's a better one that is just still in development, and using that extensively in all of our clinical trials so that the body of evidence grows quickly across multiple trials in terms of neurofilaments and other biomarkers and disease progression.
Okay. Thank you.
One additional area I'd highlight for opportunity is digital. We didn't talk much about digital, but I think the underlying premise for digital biomarkers, one is you can do it remotely, which is really important, and as we've seen in the past year and a half. But I also think that with the ability to sample more frequently in a real world environment for patients, hopefully digital tools can give you a bit with that increased sampling, a bit of increased sensitivity. And so really that you can get that earlier clinical read on fundamental properties of the patient's life, be that activities of daily living, be that as simple as movement with an accelerometer, be that reachable arm space. I mean, there are any number of different ways people are approaching digital, but I think just getting digital to a point where it's gotten the validation and we've collected the appropriate longitudinal data will be really crucial. I think there's a big opportunity in digital.
Thank you. Thank you very much for that. I think the next one, you may well have answered quite a bit of this already, so this may be a very, very short answer. So where do you see biomarkers in ALS research in the next 10 years?
So I think simply I'd say, I think Angela highlighted this, but I'd simply say that they'll be included in every trial, one. Right. And there'll be a suite of them. There'll be the simple fluid biomarkers as we work out neurofilament. We'll probably have an imaging biomarker, and we'll have some at home, we'll have some digital stuff. And so I think we'll have a compendium of biomarkers that become commonplace in trials, and they'll give us, particularly in phase 2, they'll give us these readouts that really tell us, are we on the right track or should we abandon this molecule? So I think that's really the goal I would aim to sort of have.
So move us to a much quicker response to the question. Sorry, Angela, go on.
That's okay. I was going to reiterate what you just said. We want no-go decisions to be made on more than just a change in a too small sample size of patients on the ALS-FRS. So we know we're handicapped there, but I think this breakthrough with neurofilaments and what is being done in the lab, as soon as these things are validated, we should see these as mandatory, and almost mandatory, but I think mandatory parts of every early ALS trial, so that we make the decisions early enough that we don't go down the wrong path. We're having extraordinary success on the academic side and interest here, and now we need to get these validated and into every program.
Thank you very much indeed. And I think also, I probably know the answer to the next question. What are your recommendations at this time?
So I think there it's broad inclusion.
Yeah.
One, it's probably a suite of biomarkers to the extent that programs can do that. And I do think there's a plea for doing the rather mundane but really important work around validation to make sure that the assays are appropriate and they perform appropriately, so you can draw good conclusions from those assays.
Which was definitely one of the concerns in our poll, wasn't it?
Yes.
Yeah.
Yes.
Thank you.
So I actually, it's too bad we agree this much, Toby. However, the validation piece should not be daunting. It's a necessary step. It is not a burden if it's done well and it's done by multiple groups. The speed with which validation occurred with neurofilaments is simply too slow. Neurofilaments took a decade. We're reaping the rewards now, but it's been around for a decade. We need these other options to be validated much more rapidly so they can be integrated. And in a way, they need to be almost obligatory in these trial design programs. I'm not saying they should be the primary outcome measure, but we need the data, and the larger the number of programs that have a strong biomarker component, even if it's an exploratory biomarker, the faster we're really going to get to a point where we're treating this disease effectively. Because in ALS, our partners are our patients. They're not just the recipients of our work. They're very motivated to get involved in these kind of programs, extremely motivated. And when we have these effective biomarkers, we will be able to come to conclusions with shorter trials. And I think even Toby agrees with me. The aim is to not make our trials 1.5 years long. The aim is to use biomarkers and better outcome measures to make our trials, at least the placebo control components of our trials, shorter. And I believe that biomarkers are going to help us do that.
Thank you very much indeed, Toby and Angela. I believe we may have some questions, Sydney.
Yes, that's right. All right. So thank you very much for that insightful presentation. And I've received quite a few questions from the audience, so we'll try and get through as many of those as possible. So our first question is, on slide 15, did the placebo control subject needed to collect CSF, and what phase of the clinical trial was presented on slide 15?
So this is a phase 1/2 trial, so it's a safety and PK trial primarily. But it was also designed to look at the PD endpoint of this SOD1 lowering. And it was a 3-month trial, essentially.
All right, thank you for that. And now concerning the recent Alzheimer's drug approval, it is my understanding that the approval was based on the reduction of plaques in the brain of the patients. Is this an example of an approval based on biomarker, since there wasn't strong evidence of reduced cognitive decline?
So in general, I don't work on the Alzheimer's programs, but yes, this was approved on the reduction of beta amyloid.
All right, go ahead.
Sydney, that program is an excellent example of how a biomarker helped move it along. And the PET imaging has been fundamental to that program.
All right, thank you very much. Our third question, on slide 17, the figure on the right, does it present significant reduction?
The neurofilament, this is exploratory data, and these were not powered for significant reduction. These are merely point estimates and representation of error. So they represent potential but not confirmed response.
Got it. Thank you. Our next question, tofersen trial. How are fast progressing patients defined?
So briefly, there are a number of different mutations within the subset of SOD1 ALS. And so for this trial, these patients were defined by the mutation they carried, such as the quickly progressive A5V mutation.
Okay, perfect. Our next question, A's TDP43 is the main neuropathological hallmark of ALS. What is your thinking about its measurements in the CSF through RT-QuIC or immunoinfrared approaches, as well as PET tracers?
I've been talking a lot, Angela. You want to take TDP43?
So TDP43 is a challenge. I think the closest we have right now of a marker is actually the PET tracers that they added in this group, Sydney. It does require further work and further validation, but there are definitely ligands that are being developed to try to track TDP43. So it will be a tremendous focus because most of our insights into ALS suggest that TDP43 is the primary pathological pathway in patients with ALS, not exclusively, but the predominant pathway that seems dominant in ALS.
Okay, well, I think we're going to wrap it up here. Thank you very much for those answers. And we've reached the end of our Q&A session for this webinar. But if we couldn't attend to your questions, the team at ARG may follow up with you. Or if you have further questions, you can direct them to the email address on your screen. So thank you everyone for participating in today's webinar. You'll be receiving a follow-up email from Xtalks with access to the recorded archive for this event. And a survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now I'm about to send you a link through your chat box, and you'll be able to view the recording of this event on this page. And you can also share this link with your colleagues when they register for the recording as well. Now, please join us in thanking our speakers, Ignazio di Giovanna, Toby Ferguson, and Angela Genge. We hope you found this webinar informative, and have a great day, everyone.
Thank you very much, everybody.
Thank
Thank
you.
you.
Thank you. Bye.